We conducted a study at Hospital Universitari Institut Pere Mata and the Early Intervention Service from Reus. Basically, the main aim of the study was to explore whether stress or stress-related biomarkers were associated to the risk of developing a psychotic disorder in vulnerable individuals. Those patients were individuals who were considered at risk for psychosis, also known as at-risk mental states. So we followed up these patients at our unit, and we studied a series of stress-related biomarkers, including hormones that are increased in situations of stress, such as cortisol or prolactin, as well as other proteins, such as albumin, that is lowered in situations of stress. In the next slides, I'm going to present the methods and main results of the study. This is a study that was recently published in Journal of Psychiatric Research. What we studied is two populations, 44 healthy subjects and 39 at-risk mental states. It's people who have prodromal symptoms of psychosis, and we had a clinical assessment with two scales to assess perceived stress and stressful life events with the Holmes-Rahe social readjustment scale. We also obtained a blood analysis in the morning to determine cortisol, prolactin, CRF, C-reactive protein, fibrinogen, and albumin. And we gave patients 3 salivates to obtain saliva at home and to determine cortisol levels during the awakening response. And then these patients were followed up for at least 1 year to see who of them had a risk of developing a psychosis. 29 of them did not have a psychosis transition, and 10 of them had a psychosis transition. This fits quite well with the previous proportion in the literature, that about 20 to 30% of patients develop a psychosis in 1 year. The question was, do these variables are associated with the risk of developing a psychosis? And the main result, as you can see here, is that those patients who developed a psychosis had an increased baseline perceived stress and also reported more stressful life events. In relation to plasma biomarkers, we found increased prolactin levels in those patients who developed a psychosis and lower albumin levels. And here in the figure, you can see below that those patients who developed a psychosis, those high-risk patients with a psychosis transition, had an increased cortisol secretion after awakening. And this measure has been associated with the reactivity of the HPA axis, that is also known as the stress hormone system. So these patients are really more vulnerable to stress. The main conclusion of our article is that there are stress biomarkers related to the risk of psychosis transition in vulnerable individuals. Of all stress biomarkers that we studied, we consider increased prolactin and increased cortisol after awakening and lower albumin as candidate biomarkers. Future studies have to study these 3 biomarkers because they may be involved in the pathogenesis of psychosis, as our study suggests.